Welcome to our lecture about cell transport, and this is the recap of our Gatorade investigation. As I go through the material, make sure you're filling in your virtual notebook that's starting on slide 42 and going all the way to the end until slide 55. This is exactly the same as a note sheet would be, so you're just filling in the terms that are missing and that are highlighted in the virtual notebook. All right, let's get started. All right, so your first stop and think question that you can answer directly in your virtual notebook is list one thing you learned in the Gatorade investigation. So over this past week and a half, we learned that many substances need to go in and out of the cell, whether they're nutrients, water, etc. In order to do so, they must cross what's called the cell membrane. We learned in our previous lecture that the cell membrane is made up of phospholipids, and we learned over this week that there are embedded proteins and carbohydrates in the membrane. There are two major types of transport across the cell membrane. The first is passive transport, and the second is active transport. Passive transport is the movement of substances across the cell membrane without the use of energy. And remember, when we talk about energy on a cellular level, we were referring to ATP, or adenosine triphosphate. There are three different types of passive transport that we've talked about: diffusion, osmosis, and facilitated diffusion. The first type of passive transport we'll talk about is diffusion, and diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. Remember, high to low. The difference in concentration of molecules between two or more areas of a given space is known as the concentration gradient. So for example, if we're looking at on the outside of the membrane and the inside of the membrane, that would be two different spaces. All right, so the difference in the number of, for example, glucose molecules between the outside and the inside is the concentration gradient of glucose. In passive transport, molecules move down their concentration gradient, which means that molecules are moving from a higher concentration to lower concentration. Molecules from where there are more of them to where there are less of them until equilibrium is reached. So when molecules are moving down their concentration gradient, they want to move to an area where there is less of them, and eventually they'll move to an area where they are equal on both sides of the membrane. At equilibrium, the molecules continue to move, but there is no change in concentration, which means molecules are moving in and out or across the membrane equally, so there is no change, there is no concentration gradient. And this photo to the right is just showing us diffusion. Molecules, these red dots, are moving from a high concentration to a low concentration, and eventually over time they will reach equilibrium, which means there will be an equal concentration throughout all areas. All right, here is an example of diffusion across a plasma membrane. So we're looking at the diffusion of these blue molecules. So there is a large concentration gradient to start at the beginning of our time lapse right here. There's a lot of molecules on one side and none on the other side. Over time, we see that these blue molecules are diffusing across the membrane, right? And eventually, at the end of our time scale, we can see that our molecules have reached equilibrium. So they are moving from higher concentration to lower concentration. They're diffusing across the membrane. And eventually, over time, they reach equilibrium where they are equal. These molecules are equal on both sides of the membrane. All right, the second form of transport we're talking about today is osmosis. And osmosis is the diffusion of water across the cell membrane. So similar to diffusion, now we're just talking about water. Osmosis allows for the movement of water into and out of the cell. And the net direction of water moves will depend on the concentration gradient of the solutes. All right, so in our investigation using our egg cell, we learned that the direction of osmosis depends on the type of solution that the cell is placed into. So there are three different solution types: a hypotonic solution, an isotonic solution, and a hypertonic solution. In a hypotonic solution, the concentration of solute is greater inside the cell than outside, and what happens is water moves into the cell. In an isotonic solution, the concentration of solutes is equal in and out of the cell membrane, or in and out of the cell rather, and water moves equally in and out, or in between the phospholipids of the plasma membrane. Finally, we have a hypertonic solution, which means the concentration of solutes is greater outside the cell than inside the cell, so net movement of water is out of the cell. So let's take a look at our example. We're going to use our egg cell experiment as an example, so think back to our experiment as you're going through this. Water, our tap water, which we saw that was dyed either blue or green, is a hypotonic solution, which means that the concentration of solutes is greater inside the egg cell than outside. We saw that when that occurred, when our cell was in a hypotonic solution, water moved into the cell to decrease the concentration of solutes, and as a result, our egg cell increased in size. Here we've seen these images before. This is what happens when you put a red blood cell in a hypotonic solution. We see that the cells increase in size. They swell, and some of them even begin to lyse or explode. This is what happens when we put a plant cell into a hypotonic solution. Notice that these cells do not lyse because they have a cell wall. So the cell wall keeps those cells stable, and they do not lyse, and water creates what's called a turgid cell. Up here, this is called a U-tube, and in the middle of the U-tube is a plasma membrane, or a cell membrane rather, and you can see that water, osmosis is occurring, water is moving to where the solutes are more concentrated, so you'll see that water is moving net to the right, or in the right direction of the U-tube, to decrease the concentration of solutes on the right. Let's look at some examples of hypertonic solutions. So if we're talking about our egg cell experiment, corn syrup is hypertonic to the cell, which means that the concentration of solutes is greater in or outside of the egg cell compared to inside. So the concentration of solutes in corn syrup was really, really high. We saw that water moved out of the cell to decrease the concentration of solutes, and as a result, the size of the egg decreased. Here are your same examples. So when we place our red blood cell in a hypertonic solution, water moves out of the cell, and the cell becomes shriveled and wrinkled and a lot smaller. Here is your plant cell. We notice that the cell wall remains in that square shape. However, the inside of the cell, the cytoplasm, becomes what's called plasmolyzed because all of the water left the plant cell. Finally, here's what's similar to our U-tube. We notice that our water is going to move wherever there is a hypertonic solution. So water in this case is moving to the right because the concentration of solutes is greater to the right. Lastly, we have facilitated diffusion. So the ability of a molecule to diffuse across a cell membrane depends upon its size, polarity, and charge. So small, nonpolar, and or uncharged particles can typically pass through the cell membrane. Remember, think back to our first lecture, the inside of the membrane where those phospholipid tails are located is hydrophobic, or it's nonpolar, right? So typically our small molecules, nonpolar molecules, or uncharged molecules are able to pass through pretty easily. Large and polar molecules or ions are not able to pass through the interior of the cell membrane, right? Because if it's large, it won't be able to get through very easily. Polar and nonpolar do not mix, so the inside of the cell membrane is nonpolar, so large polar molecules won't be able to make it through. And then finally, ions are charged molecules, or charged atoms, excuse me. So the nonpolar, uncharged interior of the membrane does not like ions. The only thing I will say about this is be careful, because water is a small molecule, and while it is polar, it is able to pass through the membrane, no problem. So water is the one exception to this rule. So facilitated diffusion is a type of passive transport, which means it does not require energy, and it's the transport across the cell membrane via carrier proteins to move molecules across. So in this model here, we can see that these green channels, or these green like circles in the cell, are proteins. They represent proteins, and their protein's exclusive job is to transport materials in and out of the cell that are not able to pass directly through the membrane. So an example of facilitated diffusion that we talked about in our investigation is glucose. Glucose is moved across the membrane via facilitated diffusion because it was too big to go through the phospholipids. So glucose is a really large molecule, and it's polar, so it can't go through that hydrophobic interior of the membrane. So if that's the case, glucose is able to be carried through the membrane via a carrier protein, or a transport protein. This protein shields glucose from the interior of the membrane, so it's able to pass through and go from the outside of the cell to the inside of the cell. In your evidence, you learned that one of the names of these carrier proteins is called GLUT4, and this is an image of GLUT4 that you've looked at before. So for example, this is the phospholipid bilayer. This purple channel is your GLUT4 protein, your carrier protein, and you'll notice that the concentration of glucose is higher outside the cell than it is inside the cell. So glucose will move from a high concentration outside to a lower concentration on the inside via this transport protein. Finally, ions are charged particles. So for example, these are ions below. When you think of ions, think of negative or positive charge, but you can also think of salts, When you dissolve salt in water, they become ions. So for example, if we're talking about table salt and you make a saltwater solution, when you dissolve salt in water, it becomes an ion. It becomes charged. Ions cannot pass through the membrane, so the interior of the membrane is neutral, so it has no charge. So it's very difficult for an ion to pass through because it has a charge. In order to pass through the membrane, they need to move through a channel protein, which is basically just another protein embedded in the membrane that is similar to our glucose protein. However, it is solely responsible for ions. All right, so here are our 3 types of passive transport. As a reminder, passive transport does not require energy and it involves materials, substances, or molecules moving from a high concentration to a low concentration. All right. Facilitated diffusion involves proteins, so this would be your glucose transport protein, your ion transport protein. Diffusion or osmosis do not require a transport protein. Let's now talk about active transport. So active transport is the transport of substances across the cell membrane against their concentration gradient, which does require cellular energy. So in many situations, cells will need to be able to move materials from a low concentration to a high concentration. Going from low to high is going against the concentration gradient in a cell. And this requires energy in the form of ATP. Cells cannot do this regularly. This is not a natural process. This goes against the laws of physics. So we need to spend energy. We need to use ATP in order to move molecules against their concentration gradient. So here on the right is an example of active transport. So certain protein transporters are capable of using ATP to move sodium and other charged atoms across the membrane, which means that sodium is able to be moved from a low concentration to a high concentration. What I'm about to say is beyond the scope of this class, but the nervous system, so the body system that requires you to react to stimuli and is responsible for like your your brain, those functions, really does require a lot of active transport on the cellular level. All right, so we're going to wrap up with a new idea. This is pretty simple, and I'm pretty sure you guys will be able to get it. So many substances, such as macromolecules and nutrients, so these are like big molecules, way bigger than glucose, are way too large to get through the membrane by regular transport processes. So instead of going through a channel protein or just through the membrane, they need to be transported in and out of the cell using vesicles. And we talked about vesicles earlier in the year when we were talking about cells. So vesicles move substances into the cell, and in this process, the cell membrane folds around the food or the liquid and forms a small pouch, aka a vesicle. The pouch then pinches off inside the cell to form said vesicle. So pinocytosis is the transport of liquid into the cells via vesicles, and phagocytosis is the transport of food or very large molecules into the cells via vesicles. So this is what this looks like. So when we are transporting large food particles, aka phagocytosis, or liquids, aka pinocytosis, you'll see that the cell membrane is able to kind of wrap around either that liquid or that food particle, and it's able to pinch off and form a vacuole or a vesicle. So that's able to transport these big substances into the cell. Same thing if you would like to export materials or food or molecules, the cell membrane is able to kind of engulf that vesicle coming from the inside of the cell and release the molecules out of the cell. And I think that's it. So make sure you get all your notes and submit your virtual notebook after you're done.